Co-pilot has control.
So we just reached 1,500 meters. We're still 1,000 meters from the bottom here. We're diving on a seamount north of French Frigate Shoals. So about
Seeing more things in the water here than we saw yesterday. Definitely seeing some small creatures, and I'm afraid I'm not very good at them. Looks like some small crustaceans. We had what appeared to be some type of a hydromedusa, but I'm not sure. And that was one of the larger ones that we've seen. A lot of smaller things that are basically unidentifiable, but the way they're moving certainly indicates that they're alive. And there goes something red right there. So we've got a lot of little critters in the water as we're making this descent.
Deep Discoverer,
and we have
passing
crossed,
2,000 meters.
So we
2,000 meters, approximately 500 meters off bottom.
Pilots, I'm going to snap the still camera. I want you guys get startled if you see a flash.
On your sonar. There you go.
You got sonar?
Yep. There it is, sloping slightly to your starboard. Bottom in video.
20 meters. You're at 50, I'm at 20. About a 20 degree altitude difference, 20 meter altitude difference between us. Means we're on a slope.
Okay, I can see the seafloor. We are at the seafloor now.
Bottom in sight.
And I do see some animals on the seafloor, by the way. So that's a good sign.
I'll follow you down.
And I'm seeing a fair number of animals
Can you bump my sonar out one? I don't like that I'm not seeing anything. And also bring the gain down.
So it looks like we're not going to have that that barren start to the dive as we did yesterday. It looks like we're going to have animals from the get-go, which is really nice. I can already see sponges and a variety of different corals even from this height.
Delta is 4. Came up a little bit.
Yeah, working on Z-bias.
Yeah. I'm holding. Auto heading engaged.
So I'm going to back up.
Yep.
Come down.
Okay, so I'm going to
Backing
take
up
some
towards you.
shots at some IDs
I'm
here
gonna come
because
right,
Roland still
head
wants to adjust
towards
my audio here. So I'm fairly certain that I
135.
can see Chrysogorgia
Get you
geniculata.
to come around
There's several colonies
port
that just passed
to
by.
match that. Hopefully
And
we
I
lined
saw
up
some
right where we're supposed to go.
unbranched, probably Isidids. And I
Oh,
also
yep,
saw
no
an
slack.
Isidid that I think is Acanella
Increasing
weberi.
depth, turning heading.
And I could not identify the sponges that I saw. We were too far up.
Order over current.
So this looks like a little bit more sparse grouping than further up. And right now it looks like it's a slightly different composition than the 2007 Hurl Pisces dives, which are at 1,700 meters. I do not recall Acanella weberi in that community.
Pushing forward.
So
Roger that.
we may see some transitions here on this dive, which would be, of course, quite interesting. Now we can see a number of corals coming into view. And definitely those look like Chrysogorgids as well as Isidids.
Since you had the time, as you're getting stabilized, this might be a good opportunity to look around and see what's out there.
Yeah, sure. I mean, it's a little bit like yesterday where you see a lot of rocks that appear to be loose, but when you get closer, they really are completely solidified into the substrate. Regarding the biology, I just saw Bathopathes, probably alternata. There's a different type of Isidid right in front of the screen right now. Not sure what that is.
Very
We'll have
strong
to be a little bit closer.
Yeah, we're currently here at 2,450 meters, and the bottom is just below us.
Actually.
And just a little bit, the images should be becoming crystal clear.
So I don't know if you can see this, but it looks like the current is moving from right to left, and it looks like it's reasonably strong.
Well, we originally guessed, but now that maybe it's just right on this slope. Looks like it's coming across me.
No, it's coming mostly from behind you.
Okay. Yeah, I could believe that.
I'm going to adjust your lower swing arms. I'm going to bring them out a little bit.
Yep.
I'm not about to guess what that red thing was that just shot across the screen. So these fluffy corals, sort of columnar fluffy corals, these are Chrysogorgia geniculata almost for sure.
Watch lead pilot.
Roger, pilot, go ahead.
Yeah, I think we're all set up here. You can see we're back on bottom. We're in view of Sirius. So when we first got down, it took a little while to get D2 in the view of Sirius, and now we're adjusting some swing arms for light and cameras, but we are set to go, I believe.
Roger that, and I guess what we'll do is we're going to be looking for both biological sample and a geologic sample. And so if you see a rock that doesn't look quite so cemented down here, perhaps we can possibly grab it. Mike always is interested in getting a rock at the deepest extent of the dive. We obviously would welcome
Don't
the help
turn that
in deciding what we perhaps should sample. At the moment, I don't see anything particularly unusual, but
So
Can you zoom in on the prim nose you're talking about?
excuse me?
Can you zoom in on the prim nose you were mentioning, please?
Yes, pilot, could we zoom in on that sort of one right dead center
Roger that. You got
at
it.
the moment?
from far that looked
Yeah,
like
it's-
Calyptrophora
Yeah, it's a
angularis, but
Video,
yeah,
go
it's
ahead.
sort of a feathery looking coral. That's it. Right. I don't know.
yeah.
Videos come wide.
I don't know. What do you? What do the rest of you
struggling
think?
with
Scott,
Right.
Amy, Brendan. Anybody have an opinion? I'm not so sure that's Calyptrophora angularis.
Yeah, I can't see it. It's not close enough for my view.
I
Stand
think he's
by,
setting
watch
up
lead.
a little bit to get another shot at it.
So we are at 2,457 meters.
So most of these rocks look pretty cemented, Mike, but there might be a couple that could be dislodged once we take a look at this.
Chris, don't waste your time on
ahead,
rocks
video.
like this. The area looks very heavily cemented with
Co-pilot?
manganese.
Yes, please.
I would suggest you move on unless there's something biological of interest.
Is our
Okay.
shutter?
So I think we're looking at a Chrysogorgia. Is this? Would you concur on that, Scott, and Amy, and others?
Video? Do we
I
have
would like
shutter
to request that you collect it.
up or down on this camera?
Yeah, this looks like a
Seems to be
Chrysogorgia
slow
to
to
me.
respond over here.
It's not one that I recognize. It's definitely not a primnoid. I see it's got some barnacles at the end of one of the branches there. So if there were a collection of interest, there's basically some added organisms you get. I think there might even be some hydroids at the tip of the barnacle.
Well, I'm not going to make the same mistake I made yesterday when we saw an unusual unbranched primnoid and we passed it by. I've never seen anything like this before, so I totally agree. I think we should request to collect this one.
Video, come wide, please. A little bit wider.
Please do collect those barnacles.
Okay, we'll get them. Specializing in Chrysogorgids. Has Chrysogorgia pinnata ever been recorded from the Hawaiian Islands?
It's actually my student Eric has a lot more experience. Eric Ponte with the Chrysogorgids. I don't know that there's very much known about this pinnata. You know, that image that I just
Okay,
sent you the
we'll call
reference
it good.
to
Go
is,
ahead and do
you know,
your
it's a
clipping.
deep one from up in the Gulf of Alaska, so it's at least the North Pacific. You know, there are a lot of differences. I'm wondering if, you know, this isn't as fully grown as that other specimen. This one
Pilot
doesn't
video's
quite
clear.
have the same sort of feathery pattern to the branching. It almost looks like in all cases, maybe it's just the angle we're at, that the branches are coming off one side of the main branch, which is kind of interesting.
Pilot, you copied videos
Uh,
too.
Yep.
Okay.
But
It's partial.
as
zoom,
far as I
you can
know,
stay
Chris,
or-
this would be a new record for Chrysogorgia pinnata.
Huh.
But as you know, there isn't all that much data from or observations from these depths in this area. That's the exciting thing about this cruise.
Yeah, exactly. Well, I can tell you I've never seen this before, and I would agree. It does appear like they're coming off of... I would agree that looks like they are coming off of one side. That was the original guess that it could be Calyptrophora angularis because that's a characteristic of that particular species. But as we got closer, it was pretty clear it wasn't it.
Okay.
And so this is
come
quite
wide, video.
unusual. And
Pulling wide.
if we're starting out with a very unusual species right off the bat,
broke
I
loose.
wonder what we're going to wind up seeing over the course of the rest of the day.
He thinks it might be something a little bit different. And then Tina from Russia says that they have something completely different morphologically, but identical genetically to Chrysogorgia pinnata from the Mid-Atlantic Ridge. So I think a sample might be warranted here because we have not seen this here from Hawaii. It
Can't hear you.
probably will be at least a new record,
I can now.
if not something completely different.
Yeah, this is quite exciting.
Might just have
and
to be a key grab.
also the fact that I don't know that crinoid, and I'm not so sure I've seen those scalpellid barnacles before either out here.
It's a combination of heading and vertical.
This could be a really marvelous sample. We're able to acquire it.
We'll get it for you.
Oh,
Pretty
always.
wonderful convenience that it shows up at the beginning of the dive.
Yes, it is.
Yeah, and as Chris mentioned, we're not doing the same thing we did yesterday where we saw something unusual early and then waited.
Yeah.
Good to know we can still learn, huh, Daniel?
Always.
But we only learn after making a mistake. That's the problem when you get older. I think that'll work better.
Drawer open. Real clear.
That's geniculata on there.
I know. I was adjusting something else. Opening. Getting us out farther off bottom.
So we're going into
It's going to go to
Okay.
one of the port boxes. Inside. How about putting it in? Can you do the port insides since it's a fairly small sample? Leave room on the larger box for something bigger, maybe.
Science, it looks like there's some black material
Remnants
in
from
there
yesterday's
in
sample,
the rock.
possibly.
Is that some of the remains of the rock from yesterday?
Science are asking for confirmation before he releases that you're okay with the rock fragments that are already inside.
Yes, I'm okay with them.
yeah.
Okay, standing by for drawer.
Chris, I hope somebody is cleaning those bins each day because it's not good to have debris from previous dives.
Yeah. So
Clear the arm.
the port front rock box was
Closing
completely
slowly.
clean, and this one apparently we missed the inside box from the second rock. So we'll put a biological in there today, Mike, and then we'll make sure a rock doesn't go into that same box, but all the rest of them are going to be clean.
Thanks, Chris.
Can you just put the craft there and then move shilling out, please? Yep.
So that was port inside, right?
Just
Got it.
double up.
Okay.
So for those who just tune in, we were at 2,450 meters and collected a chrysogorgid,
finish
a
close.
deep sea coral. This is very exciting in that it's the first biological specimen
closed.
ever collected by the Okeanos explorer. And we're also seeing the deepest images ever recorded inside the Kanehunamoku Marine National Monument.
Watch lead, this is Nav.
Roger, Roger Nav, go ahead.
Can I go ahead and confirm sample ID?
Right, the sample ID is definitely a Chrysogorgia, a Chrysogorgia species. Right now the species is in question, but the genus, I don't think the genus is in question, but maybe just put Chrysogorgia species ?
All right, thank you.
Yep.
Okay, co-pilot.
So these other bushy
Take it back.
corals you're seeing on the bottom look like there's some more of this Acanella weberi. Not all of them, but some of them. We see some unbranched corals as well. The stock sponge was probably a Caulophacus that we saw earlier during the collection event.
Okay.
Chris, could we zoom in on some of those squirrels? Just 'cause we haven't been at these.
065, 2 times 25 meters.
Okay, video. Let's zoom in on this coral top center.
Good.
Co-pilot, can you tilt up my camera? There we go. Okay. Watch lead, is this one of the branching ones you're interested in?
I'm trying to figure that out. If we could go down a little bit further toward the base.
Okay.
Take
Tilt
a
down,
look.
co-pilot.
And maybe zoom in right where the branches come out.
Okay, I'm just going to spin over a little.
Oh, that looks different.
Is that ship move underway? Great. Nav, is that ship
Looks
move
like there's
underway?
been some damage on the branch on the left, and it's perhaps regrowing and regrowing downwards to join the lower part of the axis.
Drawing
Right in
now.
about
I
the
was
middle.
just updating John.
Yeah, I see that. Interesting.
Got it. Bridge, this is Nav. Hi, can I get a ship move of 25 meters at 065 degrees at 0.2 knots?
It's a little difficult to tell whether it's.
An internodal brancher.
I think it is
Internodal
too.
brancher, then it wouldn't be a canella. Maybe not. Maybe it's branching right just above the nose
Sorry,
there.
did you hear that?
Yeah, it's a little unclear. The branch on the dead
yes,
branch
can I
on
get
the right hand
25
side
ship
looks
move
like
of
it's
25
internodal,
meters
but the
at
one that's still
065
alive there
degrees
looks like
at
there could be a
0.2
node where
knots.
it's coming out. But the interval's a little weird in that it would be a very short interval right there. So how about a pilot, could we zoom in on the coral colony to the upper left, that other one?
Yeah. Just readjust a little.
Sure. Right. Perhaps that'll be a little more representative of the others.
Okay, co-pilot, can you tilt up?
Tilting up.
And there's a few requests here to turn the lasers on again.
Okay, you're free to zoom.
Yeah, these two look different than everything that's in the background there.
Can you get lasers too, co-pilot?
Thanks.
See a nice ophiuroid there hanging on.
Yeah, maybe a little bit closer.
Okay, full zoom video.
So I'm interpreting this as internodal. I don't know if anyone else has a different opinion, which would definitely make it not an Acanella weberi.
Oh, losing it.
I agree. That's internodal branching.
Yeah, I think
So not a canella. It's also too planar. There's no multiple branches coming out in a whirl. So
Right.
that would, at least for the time being, be classified in the Keratoisis genus.
Okay, you can go back in
Right.
video.
Yeah, I think this is perhaps just a small possible keratoisis. And I agree, there isn't that whirl of branches coming out of the base. So
Ship moves underway.
some of those that are further up slope, they may have that whirl. When he zooms out a bit, we'll take a look.
Losing it.
And you can
Going
see
back.
some of the manganese crust botryoidal patterns on the rocks very clearly right there as he was panning over the rocks. that sort of bumpy texture.
That's pretty.
Yeah. Yeah.
Camera seems great today.
Yeah.
Tilt down.
He was panning over the rocks. That sort of bumpy texture.
That's pretty.
Yeah. Yeah.
Camera seems great today.
Yeah.
Tilt down.
So I think we, once video is satisfied, I think we're okay on this one, and maybe we can potentially move up slope a little bit and look at some of those other ones. And then
You can also see there on the lower branch on the right that a group of small barnacles has taken advantage of the tissue that's been removed from the bamboo coral skeleton. It's a good settlement
yeah.
spot.
See those?
Yeah, see they're all stretched out there.
Great.
Interesting that they were all facing in the same direction.
Yep. And you see the crinoid on the very top of that center branch?
Yeah, let's tilt up a
Yeah.
bit. Yeah, that's pretty good. Yeah. There it is.
Yeah.
Didn't hold that for 12.
So I'm assuming
All
that
right,
this
let's
is.
go wide. I think they got it. Yeah.
Yeah, I'm assuming that that's
Yeah.
some species of Kamehameha. It's a
Is it whipping down there?
stalkless
Yeah.
crinoid.
Jeez.
Okay.
So
While we're looking around here, Chris, I can share with you, I'm looking at the paper by Steve Cairns from 2007 where he described Chrysogorgia pinnata.
Even my heading.
And of the 59 species of Chrysogorgia, that is the only species that has that pinnate branching.
Maybe you should do another 20
So
or
if this is
something.
not Chrysogorgia pinnata, then it is a new species that we just collected. So that would be
Yeah,
interesting.
you're pulling me off.
Um,
Gotta get a lot closer.
Chrysogorgia pinnata is only known from seamounts off California and Oregon, apparently between 1,968 meters and 3,275 meters, but most of them are between 2,600 and 3,100.
Can we add
So
another 20 meters to that
interesting
move?
to compare when we see that sample later.
yeah, absolutely, and that's I appreciate you looking up that information. And again, I assume that if it does turn out to be pinnata, it's going to be a new record for the Hawaiian area. Is that correct?
Absolutely. So far, it's only known from off of California and Oregon. It was only described in 2007, so this would be one of the only other samples collected, apparently. The holotype was kind of damaged when it was collected. And also it notes that mostly it's been found at temperatures between 1.6 and 1.9 degrees Celsius, which makes me ask the question, what's the temperature right here? Do we know?
We're at
Yes,
1
I
point.
see 1.7 is on the...
Yep.
Thank you, now.
2007 where he described Chrysogorgia pinnata.
Even my heading.
And of the 59 species of Chrysogorgia, that is the only species that has that pinnate branching.
Maybe you should do another 20
So
or
if this is
something.
not Chrysogorgia pinnata, then it is a new species that we just collected. So that would be
Yeah,
interesting.
you're pulling me off. Gotta get a lot closer.
Chrysogorgia pinnata is only known from seamounts off California and Oregon, apparently between 1,968 meters and 3,275 meters, but most of them are between 2,600 and 3,100.
Can we add
So
another 20 meters to that
interesting
move?
to compare when we see that sample later.
Yeah, absolutely, and that's I appreciate you looking up that information. And again, I assume that if it does turn out to be pinnata, it's going to be a new record for the Hawaiian area. Is that correct?
Absolutely. So far, it's only known from off of California and Oregon. It was only described in 2007, so this would be one of the only other samples collected, apparently. The holotype was kind of damaged when it was collected. And also it notes that mostly it's been found at temperatures between 1.6 and 1.9 degrees Celsius, which makes me ask the question, what's the temperature right here? Do we know?
We're at
Yes,
1
Oh, I
point.
see 1.7 is on the...
Yep.
Thank you, Nav.
Okay, well, we should probably make some movement since I think we haven't, we've been so fascinated with the spot, we haven't,
Of course.
haven't
So
covered
what's the general plan is to move south up the slope?
that's correct.
Okay. We just put in a 45-meter move for Sirius to get her over D2.
Great.
And then we'll be all lined up and we'll proceed south.
So while we're waiting on that, we're looking at some of these other bushy corals, and again, they do not seem to have the same whorl type of structure that we saw yesterday on the Acanella weberi. So I'm assuming that these are all different, and perhaps something maybe in the Keratoisis genus. And there may be...
They're also all oriented, sort of leaning over in the same direction to the left. It suggests the dominant current is moving across from right to left.
Yes, and actually in 2007, that was the direction of the current as well. So,
Co-pilot, where do you think the local uphill is? You can just keep following local. About 45 degrees to your port.
Yeah, which is very
So what
local.
Oh, that's too local.
wrong
Okay,
direction.
yeah.
Yeah.
So we'll just go, we'll just go southwest. It won't quite be uphill.
Yeah, so if you want to start...
So do you mean
sidling.
southwest or southeast, pilot? Because it looks like
sorry,
way
I meant southeast.
Oh, okay. Yeah, thank you.
Good catch. We give the bridge numbers anyway. It's harder to get those backwards.
Right.
So yeah, I don't know if you can see yourself in my fisheye,
Just to the side, yeah.
but you're, well,
So I have to come just
you're-
west, right?
you're 045 right now, basically.
Yeah.
Is that the way you're facing? You're facing, no, you're facing
Yeah.
due east. Okay.
But I can just sidle to my starboard and I'll end up
Yeah.
under you.
Yep.
Okay.
So if you'll just start doing that, and you got yourself nicely planted for a while there, huh? Seems really high for you guys. Holy cow.
Yeah, this is what it was yesterday.
So I think what we should do is we'll go ahead and survey up a little
How's
while,
this?
keeping an eye open for rocks and potential new species. We can still collect one more biological
Not getting tugged as much.
and
Still a
start
little.
seeing what the extent of this
Nav, can you zoom
bed
in
is
all the
right
way
here.
in high pack to show the ship and the two vehicles? We like to keep it zoomed in to watch where we are.
Okay, I'm starting to see your light pool.
Okay.
I'm going to pick up a little. Just a touch to give you some room. You're going to keep dropping as you go under me, so you don't need much room.
Yeah.
So we may have another Isidella trichotoma over here to the right. That's based purely on its planar structure right now.
Watch lead will be a bit high as we go down this local high, because we can't really face
It's
uphill.
definitely an isidid.
And we did see some of these yesterday as well.
Okay, I should start to see you soon. Make sure.
Oh, you were looking straight out. Okay, I'll go forward.
There you are. And I'm still facing, well, actually a little, maybe there's a strong current up here that's pushing me off now, because you're right under me. Still feeling it, so.
Our delta is 22.
Yeah, so I think our next move is going to be something.
And we have an Iridogorgia
150,
coming up
so
on
let's
the upper
start
right
to inch
corner.
that way.
And looks like Iridogorgia magnispiralis from
See which
here, a
direction
relatively
you wanna
small
sort of
one.
head off in. You can see it's still sort of a little to your starboard.
And that's the first Iridogorgia that we've seen on this particular cruise. Hopefully we'll see some more because they're quite gorgeous animals as we get upslope.
Okay, pushing ahead and centering.
It's just my guess on the direction we want to go. Nav may have to a little more.
And as we're continuing along here, there's an October sponge to the upper right. And this is going to be a Tritopleura.
That was not me.
So I'm
We've
not
never
sure
been
which
here.
species, probably Tritopleura species 1A at the moment. And that's what it's known as. Got some more of these unusual chrysogorgids that we already collected.
Okay, I
A
think
few
you can come
comments
down a
about
lot.
the
Our
rocks.
delta's
It looks
24.
like the slope has got quite a lot of rubble, and then it looks like there's a little bit less directly in front of us. The slope to the left, that is. Like we've got a little more of a solidified substrate that we're going over now.
Okay, Nav. We're just about in a good marching order for 150. Does that
And
about
we've got
match the direction
a
you
number
think
of
we should head?
unbranched bamboo corals.
All right.
You just had a squat lobster Munidopsis.
Did
Can do
yeah?
a-
Well, we must have passed it already.
do
We'll see if we can
at this point.
see one a little bit later on.
100 meter
I'm
jump.
sure there's probably a lot of them around. Looks like we have some type
Doing 100,
of
let's just do 0.2.
possible
steady.
Iridogorgia or Chrysogorgia there to the right, and a fairly large planar colony of something beyond it.
Chris, as you cruise along this cliff or area, there might be little landslides where loose rocks might be available.
So would some of these rocks be suitable? I was thinking you were saying that this wasn't a very good rock hunting area earlier on.
Oh, that's correct. Previously, you were on a manganese encrusted area. Well, this is probably too, but where there's steeper sections, they may break off debris, and you might be able to get a sample. This area we're settling on now, though, I don't think you're likely to get a sample.
Right.
But
Okay.
maybe the pilots.
Okay, video, let's go in on this bushy
The
coral.
key thing is at the base of a steeper scarp, things fall off, and that might be the best area.
Yeah,
Video in on
no,
this coral.
got a couple comments here. Scott France doesn't think it's Metallogorgia, and I don't think so either. It's too large of a plume. It looks like some very strange Chrysogorgia.
Looks like the main axis is ever so slightly spiraling.
Turning slightly to starboard.
But it's got a lot of sub-branching, Scott, and you still think it'd be an Iridogorgia with all that sub-branching?
That's a very good point. This would be very interesting, but my student Eric Ponta and I described a species, a new genus, Pseudochrysogorgia, that looks like a combination between Metallogorgia and Chrysogorgia. So this could fit that bill. It's only known from the area of New Caledonia, so this would be interesting.
So do we want to go with two Chrysogorgias or possibly wait a little bit and see if there's something else? This, I think, is going to be a very target-rich environment today. And if you...
I wasn't advocating to collect it. I was just making the observation.
Sure. Could we pan left a little bit, pilot, or video?
Pilot pans. Video zooms.
So there is a very unusual sponge here.
Is it within on
And
this
there
rock?
he
or-
is. He's
I
on that
see.
little
Yes.
stalk. He's just to the left of the
Got him.
branching coral. It's just to the left of the
Yeah, I can
lasers
take
right
camera
now,
with
that
you.
very small toothbrush sponge.
Uh-huh. Okay. You're free to zoom. Oh, we should probably stop this move as we're...
What
Well,
are you gonna sample?
So
watch lead, any chance
that's
of
very
sampling
bizarre.
this?
And I'm going to ask Amy here whether this looks like the
Well, just stop
unusual
anyway.
sponge she collected in 2003 off of Pioneer
started
Ridge,
feeling
which
that
turned
move
out to
yet,
be a
so-
demo sponge.
So yeah, you'll go a little...
I've seen the museum specimen that Steve Cairns sent me, and it doesn't really look like this. I have absolutely never seen anything like this, and I saw one earlier as well.
Just tweaking starboard.
I feel like I've seen it before, but I don't remember the ID on it.
Yeah, you collected a small one. I wonder if I could bring up his... I bet I don't have it with
Okay.
me here.
Yeah, so co-pilot, you might pass a little over me, but
I'll,
we'll be
I'll
safe.
look through my collection sheet
Then
and
we
get
can
back
just
to you on
start
it.
again when they're ready.
Okay. Well, again, maybe we'll pass on this, but it's a gorgeous imagery of this sponge. So, and it's quite unusual. I'm sort of keeping our options open here. You know, this is going to be difficult to collect, number one, and that's probably going to be our last collection if we grab this thing. So maybe we'll just hold off for the moment.
And there is a request to zoom in on that sea pen that's just...
And
This
there he
rock,
is. He's
I
on that
see.
little
Yes.
stalk. He's just to the left of the
Got it.
branching coral. It's just to the left of the
Yeah, I can
lasers
take
right
camera
now,
with
that
you.
very small toothbrush sponge.
Uh-huh. Okay. You're free to zoom. Oh, we should probably stop this move as we're...
What
Well,
are you gonna sample?
So
watch lead, any chance
that's
of
very
sampling
bizarre.
this?
And I'm going to ask Amy here whether this looks like the
Well, just stop
unusual
anyway.
sponge she collected in 2003 off of Pioneer
started
Ridge,
feeling
which
that
turned
move
out
yet,
to be a
so-
demo sponge.
So yeah, you'll go a little...
I've seen the museum specimen that Steve Cairns sent me, and it doesn't really look like this. I have absolutely never seen anything like this, and I saw one earlier as well.
Just tweaking starboard.
I feel like I've seen it before, but I don't remember the ID on it.
Yeah, you collected a small one. I wonder if I could bring up his... I bet I don't have it with
Okay.
me here.
Yeah, so co-pilot, you might pass a little over me, but we'll
I'll,
be
I'll
safe.
look through my collection sheet
Then
and
we
get
can
back
just
to you on
start
it.
again when they're ready.
Okay. Well, again, maybe we'll pass on this, but it's a gorgeous imagery of this sponge. So, and it's quite unusual. I'm sort of keeping our options open here. You know, this is going to be difficult to collect, number one, and that's probably going to be our last collection if we grab this thing. So maybe we'll just hold off for the moment.
And there is a request to zoom in on that sea pen that's just to the bottom of the screen right now.
Right, so the
Gotcha.
red stalked object at the base of the
Tilting down.
boulder, it's dead center.
Slow moves. Yep.
Yeah. So Andrea, this is your request. Do you have any ideas here? I've got about three possible
bit
families
right.
this could be in.
That was all.
I think you would need to be able to see the sclerites to be sure to distinguish between things like Anthophyllum and Portillum or even Copobelemnon. So this is probably going to remain just a sea pen, rock pen mystery.
Yeah, I would agree.
Right a little.
And these animals are called rock pens because of the unusual anchor at the foot of them, which is more designed to attach to rocks than most sea pens, which tend to have sort of a hook-shaped anchor, and it goes into the sediment. So this is
Consensus by Andrea and Tina seems to be Anthophyllum.
yeah,
certainly a reasonable choice.
yeah. The other possibility is that it could be a scleraptilid, and maybe even in the genus Calibelemnon, but it's certainly not the same as the shallow water Calibelemnon that we've recorded here at about 400 meters. I think those would be the two best candidates.
So we're just going a long contour here.
Not at all. Yeah, no problem. So we've got a nice variety of isidids here. We've also got some chrysogorgids, some that are a bit perplexing to us, and we collected one of them. And this is a moderately dense bed, so it's pretty exciting to actually see this number of animals already at this depth, which is well below the other bed that was discovered. But I can tell you right now that the composition of this bed is completely different than further upslope. So, wow. So we definitely had a transition, even if the bed did, even if we did, even if the high density community continued downslope to this depth, it clearly transitioned into different species, which is also very valuable to know. It clearly transitioned into different species, which is also very valuable to know.
And that is one of the main questions that we're trying to answer through these expeditions here.
Co-pilot?
Is what's the depth
Looking
range
good.
of some of these species, and what are the environmental factors controlling
Come up
the
to
depth
your port,
distribution of
look
some of these
uphill a little
organisms?
maybe.
Yeah. The current's uphill at this point, so I guess I'm overcompensating. It's not as strong as we thought, though.
Yeah.
It's been described
What's
in
that?
the world
Did you
so
say you're
far,
moving?
and if I'm not mistaken, it was somewhere on the order of
Very slightly.
5 meters
Okay.
long.
It's hard to tell
Yeah,
because
that
the
is
contour is
correct.
on my
So
port, I
that
don't have
one we
anything
just
to
went over is
compare.
just a little
Just slide
baby
along, yeah.
compared to the one that he
anything to
described.
compare to.
So the sort of golden colored animal that we see just going off screen is most likely sea penned in the genus Holypterus. That would be my best guess from this view, but I could be completely wrong.
Yeah, let's zoom in on this because this might be a
Yeah,
black, and
yeah.
if that is a black, then we'd like to collect it.
Yeah, maybe, pilot, we could zoom in on that orange colored columnar,
Okay.
sort of feathery type of colony.
Copy that. Settling in
If that's
here.
alternata, that's the biggest, longest alternata I've ever seen.
No, that looks
Oh,
that looks more like Parantipathis.
Okay, video. I think we're fairly stable.
Well, let's wait on the.
Well,
Hi, Chris.
yeah.
This is a black coral. Looks a lot like Parantipathes, the genus Parantipathes.
Yeah, I agree with you, Scott. Here.
Yeah, I think this, I hate to say this, but Dennis Apresco provided some funding for this cruise with the agreement that we would collect a couple of blacks, and this is a very unusual black coral here. We have a possible species of Parantipathes in our gallery, but I don't think I don't think the genus is confirmed here. Is that correct?
That is correct. There's one
sounds like we're
specimen at the
sampling. We should probably
Smithsonian
stop the
that is very, very butchered.
Sampling this?
It was collected by trawls.
Okay.
So yeah, this is definitely a collectible because it is likely a new species.
Yeah, so my initial guess said Lipterus, the sea pen from a distance, was way off, obviously. And so, pilot, I think we are going to go ahead and collect if we don't have any strong objections from others in the ECCs.
Okay.
This is definitely going to be something new, probably a new record at the very least. Pretty good multi-beam imagery for this area, so we had already decided on a path straight up through the ridge, and that's what we were going. We're sort of in a little trough as we go back up the ridge, and I think basically what we'd see on Go ahead.
I was just going to say, the reason I ask is so you might find a wall that there could be talus at the base of that would be suitable for picking up rocks. The surface you're going across is heavily cemented, and there'll be no loose rock.
Right.
Okay. 150 for me. Copy that. Oh, 155. Jump past a little.
Well, I guess the main thing, Mike, is that we would like to try to get a little further up and get a little bit of shallower depth underneath us, and maybe we can take a look for possible walls. I need to probably bring up the image of the site here and see if there's any good-looking adjustments to the path here that might fit the bill for you.
Co-pilot, once you're settled, can you maybe fiddle with the lights and see where that glare is coming from?
Oh, yeah.
We're very flexible on the rocks, Chris. It just you seem to be drifting laterally rather than moving up the hill.
Yeah, I think what we're doing here again is we're trying to get all the vehicles lined up here, the ship and the Cirrus and the ROV before we proceed. The ship's got to move a certain amount of distance before the ROV can proceed.
So actually what you're seeing is the actual path between waypoint 1 and the cliff it doesn't show up on multi-beam, but we do have to move laterally along.
Oh, I see.
We did. It was a local hill that wasn't even in the multi-beam.
Oh, okay.
So that's why we're moving laterally. Now we're actually facing 155, well, 116, but now we're starting to actually face uphill. We're coming to the rise that will lead to the cliff, I believe.
Oh, good. Okay,
yeah.
Great. Thank you. I'm going to pull up what do you think about these? They're potentially easy grabs, but would this suit the bill, or would you prefer to look for something else?
They look like they may be cemented.
It's always bad to be greedy, so I'd rather have a rock in the box than nothing. But are these rocks actually loose?
No, they don't look loose, but if we did drive around and find something, we could try.
Yeah, how about that one that's sort of propped on top of the other one? It looks like it could be a little bit loose there. Like there's
Yeah, absolutely.
Is that too big, or would that be suitable, Mike? That one to the upper, slightly to the right of the screen.
I can't tell what you.
I see what he's talking
So
about.
it's the laser.
I haven't figured out what you're talking about yet.
It's the one to the right of the lasers right now, and then slightly below and to the right of the laser. It may be a little too big.
Oh, that was too
Well,
big.
I like big
Let's go
rocks.
now.
Yeah, it was too big, Mike. Pilot confirmed.
Okay, well, we will look
Yeah.
for the, as you said, the small cliffs that might have rubble underneath.
Yeah, let's go ahead and make some tracks here. I think this
Okay.
looks like a yesterday scenario where we looked around and they were all cemented in until we finally found a decent one.
Okay. So the move has gone pretty far, so it should be moving along.
Okay.
What's the speed of the move? Okay. We could go up to 0.3. Okay.
We're at 2,480 meters. We're about 2 hours into our dive. So far, we're very excited. We've collected 2 biological samples. One that looked like a Chrysogorgia, and also more recently a black coral, Parantopathes. And we're currently moving along, looking to collect a geological sample. There's some nice rocks in this area, and we're looking for something that might be loose for us to grab. The most interesting thing is the abundance here. We see quite abundant bed, and not as abundant as the organisms or as dense as what we'd seen in a shallower dive that was conducted by a manned submersible in this area in about 1,700 meters, but still we see a high abundance of corals and sponges.
So, pilot, is it possible to increase the speed a little bit?
Yes, we did just increase from 0.2 to 0.3.
Is it possible to go to maybe 0.4? Would that work, or would that cause problems?
I
Yeah.
think this is a good speed. It does take a while to get Sirius moving, so that is part of the delay.
Okay.
So you'll see once Sirius is moving at 0.3, it's fairly quick.
Okay. Yeah.
And then if we start going too fast, it takes forever to slow Sirius down.
I see.
It's kind of it's a dance.
It's a good way to describe it, the dance.
But I agree. After the sample, you call in a move. It takes about 10 minutes to get Sirius moving at this depth.
I see. Okay. Right.
Go ahead,
Yeah.
video.
And Bruce Mundy, our local deep sea fish expert here in Hawaii, just made a comment on the chat that there is abundant corals in this area, but unfortunately no fish so far. And yeah, we can confirm that here in the control room that we have not seen any fish on this dive. A few shrimp, but other than that, nothing that's moving.
A close-up of this one if we're right in front of it.
Sure. Video, I'll just center this up. Do a zoom.
So I'm not so sure this is Isadella. I guess I'd like to get a little different view. It certainly could be, but also be something else.
Okay, how's that look? Lots of associates.
So this is going to be in the family Primnoidae, and this looks like it's sort of a classic example of Calyptophora angularis. That would be my best guess. Beautiful colony. And it's got a bunch of things growing on it.
Oh, just rose up a little.
Yeah, I just took a look at the chat room, so there's been some discussion, and looks like everybody's on board that it's a primnoid. And got a bunch of some type of anemone that's growing on some of the stems.
You want to tilt up with the camera, or... Oh, okay. You're right. I will lose the bottom eventually. Oh, that wasn't very smooth. I think that works better when you set down on sand and then slowly take off.
Yeah. That, right above it, just a little snap. There's this little critter crawling on that rock. It's really cool. Down just a hair from there, just off the tips of the coral.
To the right a little?
To the right a little. You can see a little white line.
Okay, want to go in video?
I guess it's hidden back behind there now.
Oh. Okay.
Oh
well, I should probably get moving, huh?
Wanted to see what it was.
All right, thank you, video. Great.
So we're now getting into...
Nice.
So Mike's looking at these flows, so I guess as we go up through these pillow flows a little further, if we could see anything that appears loose. But again, we gotta make some distance here. And everything looks pretty solid right now to me.
Yep.
These are gorgeous pillows.
Nice over to Gorgia there.
Zip tie is in a bad spot. There we go. That's better.
Yep, and another Chrysogorgia geniculata, and some more some of these unusual chrysogorgids and isidids.
How's the ship move doing now?
It's been stopped for a while. It looks
stopped. Okay. So how far is D2 now from the first contour on multi-beam? Okay, so we could call in another 50-meter move and just keep leapfrogging. Sure.
None of these look loose, do they? No, that works.
They're all giant.
Slowing.
It's another one of these unusual chrysogorgids. Let's see, what does Scott say? It looks like another of the chrysogorgia, pseudo-chrysogorgia, unusual ones that we saw. She's looking at the red object, the commensal on it.
Settling, but we're there. Come on in. Yep.
It's just a little crab, Mary, something you wouldn't be interested in. Just joking, of course. Any ideas, Mary? Can't really see the front of the carapace yet.
I just lost your picture.
Oh, you lost the picture. That was bad timing. We're looking at the underside of it anyway. You can't really see if there are any spines on the top of it because he's sort of upside down.
Well, I can tell you what family it is. It's the Chirostylidae, and it's probably a genus related to Eurotychus, which is a group I'm studying right now.
Ah, I see.
I don't think you can collect it, but try to get a few pictures, please.
Sure, there. You've lost the video, but they've got a lot of close-ups right now they're doing.
So, Jeff, I just have serious note.
Yeah, Mary, looks like it's got very broad spine straight out the center between the eyes, and then looks like it's got some type of anemones or something growing off of it, unless it's antenna on this thing.
This coral's actually moving around more than you would expect a coral to move.
Yeah,
Current crop wash going on.
yeah, I guess they're
So,
antennae. But they've
pass
got
you by.
they're very frilly antennae,
Yeah, very feathery.
very feathery.
Video when you're ready, I have to jump off.
No real.
Feathery antennae are chemosensors.
Huh.
They almost mimic the polyps there.
Right, and they've got a lot of hairs on the ends of the ends of the claws. Okay.
Good crab.
Got another Ritogorgia ahead and a Chrysogorgia, and we got a sponge coming up, which is Tritopleura. And this is probably a species B. Still are showing lots of bamboos all over the place.
Interesting crinoid, possibly. Video if you just want to snap in real quick, just from here. I'm not going to settle, just trying to catch up to the ship. Go ahead. Snap in video. Before I lose it.
Nice crinoid.
Just hanging out.
Yes, and that is a very interesting crinoid.
Alright, video wide. Continuing on.
In fact, I'd say I have not seen that species of crinoid here before.
Would you like a better look?
Got a...
Nope, we're going to look at these two species. Then we'll move on.
And Nicole Morgan just is asking whether the crinoid might be Ceramitra tricerealis.
Yeah, well, Nicole, you got me. I don't know that species or even that genus. So it's got 3, 4, 5, 6, 7, 8, 9 or 10 arms.
Okay, go on in. Yep.
Found in these pillows that just auto head
Got,
and just lock down a little. We'll kind of hook the front end. Just give it itself.
So I'm looking at the fact that it's got bare filaments right at the end. So this could be a telecrinus, but the ciria at the base look a little bit differently. And so a telecrinus is characterized by having a bare filament at the end of each of the arms. So, but usually they're longer and finer than this in my experience. So if this is a telecrinus, it's something very different. And do we have any other discussion in the chat room? Diva's got Comachalid.
Just going to touch down a little.
Now that is spectacular video. And Astrid asked whether there was copepod parasites on the crinoid's arms, and they're really not sure.
I don't see anything on the arms. Maybe something right here. Let me tilt up just a touch. Go ahead, video.
Yeah, she's talking about those little white things, yeah.
There's one. I see a video you're talking about, yeah, right there.
Yeah, definitely copepods.
Is that full zoom? Yeah. Thanks.
Okay, so when we're done here, there was a coral colony that's a little bit up. I don't know if it's possible to get over there and take a look at it.
Yeah, it certainly is. The ship is actually stopped at this point, so
I'm looking down.
Good.
A second.
No danger.
You let me know if I need to go, Carl. This one here?
Yeah, this one here.
Yeah.
Okay, I'm gonna settle in.
And Nicole Morgan says that the crinoid looks very similar to the one we've seen that they sampled in on Necker Ridge, which is to the west of us a bit.
Okay, video, come on in.
So that one that you sampled, Nicole, is that the ID on it, that Ceramitra tricerealis? Okay, this is Chrysogorgia, is that correct? This is clearly a different Chrysogorgia. I can't really tell whether it's planar, but it could be Chrysogorgia chryses. And if it's a bushier, it could be Chrysogorgia stellata. Not really certain. Or something completely different.
The light might be a little better up here. Let's try that. Okay.
So, Scott, you want to go out on a limb and try to come to a genus? Chryses is the, and that pun intended there, and Chrysogorgia crises is the one planar one that we have out in Hawaii. And while Scott is pondering whether he's going to take the leap, Nicola's weighed in and said Chuck Messing has
I need to catch up.
indeed ID'd the similar crinoid they collected off of Necker Ridge as that. Now it's off screen. That's Sumametra, something like that.
1-5.
What else going on with that? Oh, my auto heading's still on. That's why I couldn't figure out what's going on with this.
It looks like a younger one, like
right.
the one in the back.
Yeah, it's definitely branching in the internode. So yeah, it's probably going to be in that group that we're currently calling Corrado ISIS.
Going a little farther video.
Workshop Copilot.
Yeah, Corrado ISIS.
Yeah, so we sort of missed that, right? There was something that... Oh, okay.
I was just on the periphery and we passed it rather quick.
Oh, I see. And Astrid was asking about sponges, and
Now
sponges
I'm
are
from the tether.
a little...
That,
Yeah, sorry, Scott, go ahead.
Could you drop a little, Copilot? I'm just getting a lot of tugs. I don't know if you have room to come down.
Go ahead, you broke up there. Are you still there, Scott, or did we lose you?
I'm still here, but you...
Is this feathery one something we've seen, watch lead?
Yes, we have seen this one.
Okay.
So yeah, let's go ahead and keep moving if we could.
Oh yes.
Thank you.
Keeping the moves dialed in.
Yep.
I'm just too fast for the ship. I puppied off.
Did you puppy off?
A little. Still tugging. Just trying to keep something new and interesting in view. Any of this look like rubble that we might want to try to grab?
Mike, what do you think about the stuff that's showing up right here?
It's all cemented down. Angle you're looking at 45. I'm way out there.
Yeah.
You could just drop a little more.
Okay.
Thanks.
Looks like some of these might be collectibles if these are satisfactory, because it looks like they could be a little bit looser on the bottom.
Hey Chris, this is Jonathan. These are looking pretty good right now. If you guys find a loose one, go ahead and snag it up.
Assume you copied, Pilot?
I did copy.
Yeah, great.
Do you want to do what Dave Wright did and find some 10 centimeter ones and nudge them around with the work skid?
The skid. Okay. This is
why don't you pause here?
Looking at that, I don't really know what is all in between there. It could be dead coral material too.
Yeah.
It could be something else. Could be
touching down in the middle skid. You can't see it.
Yeah.
It's just so flat here.
I mean, they look pretty concreted to me, actually.
They do. Okay. This one back here, I'll show you the one that moved. That guy right there moved. I bumped last. See if I can get it to do it again. How's the move going?
Sears is catching up to you. I'll just spin around to get you in camera view.
See that one move? Right there.
Yeah, it
Yeah.
moved.
That's the one I saw move before. Is
that a collectible,
let's get the
Jonathan or
lasers
Mike?
and see how big it is before.
Well, that one's too big.
Which one are you referring to there, Chris?
The one that's now at the bottom of the screen. It may be a bit big, but
right.
The one that you just kind of tapped a little bit, where
the
floating sediment
so
is that?
Yeah, the lasers are on it right now.
It's 50 centimeters, so that's like.
What's the actual size limit to what we can grab?
Loose,
yeah, that one I think is going to be a little too big.
I was hoping that some of these ones nearby might be equally
Don't worry
loose.
about that, Chris.
Right.
The pilot will tell him.
Yeah, it might be.
Awesome.
I'm just gonna.
I think some of these smaller ones might be loose.
I'm gonna just reach out and give it a nudge with the fingers. We'll tap it. If it doesn't wiggle, we'll give up.
Okay.
Try with the Orion if you're concerned. The craft. But a couple of these definitely look like they're could be loose. Just getting a less awkward toe hold on a front skid.
So we're currently at a depth of 2,445 meters. Getting positioned to see if we can collect one of these manganese-crusted rocks.
Yeah, I might.
About that one over on the right.
It's a cool looking coral.
Very cool looking coral.
Okay, I'm headed 150 again.
Okay. Well, I don't think that any of this is going to be worth a nudge. It all looks pretty well cemented, and we're on a hill, so it'll roll down if it comes free.
Yeah, and we're back into some pillow formations, I can see.
Yeah, I think if you look at HiPAC, we've come to the steep part of the terrain. And it's definitely reflected in the topography here.
Maybe something fell off this big piece. We can look below it.
Yeah.
If you see anything loose looking down there, I'll set down a little more.
Chris, there might be some samples that have fallen off of this wall of pillows right here that we might be able to pick up.
Yeah, he's settling down and gonna take a look. Right now, though, it's looking pretty cemented.
Yeah,
but
anything back in there that looks loose?
Oh, hey, so you might see
the
one.
Lasers. Is there anything in there? Is that all?
Oh, I see that one. You mean below the lasers? Maybe.
Co-pilot? No. Co-pilot's shaking his head. He doesn't think so.
Oh, okay.
We have an experienced pillow lava picker-upper next to me here. He says he's tried probably 100 different pillow lava grabs and failed a lot of them.
A dozen.
He knows what not to look for.
Okay.
Is the story going to grow to 1,000?
Yep. Well, I'm trying to make your credentials look better.
Yeah. All right, I'm going to keep coming up here as you come up this.
Yeah, just a pretty nose.
Yeah, this is the first time we have some three-dimensional stuff to look at.
It's like another one of these. Is that what they were calling Thelrilla right there?
No.
That wasn't okay, because that looks like maybe a geniculata.
Geniculata, yeah,
right.
definitely.
Okay. Darn it! I'm sorry I missed that. It would have been interesting to see it. So there's another Ritagorgia.
Yeah, 170.
Here from the monument.
Nav, co-pilot. Could Should we do a 30-meter move? Bearing
Yeah, it is very
170,
interesting, and there's
0.2
been
knots?
some comments on the chat about the lack of fish and the scarcity of sponges on today's dive. We have not seen any fish.
Still a little deep for seeing a lot of fish, isn't it?
Yeah, pilot, maybe if we see a pile of these little pillows that are raised up so that the skid would hit them
So
first.
there
we
is
could
a
just
question here in the chat about the
nudge them.
current, and yes, there has been a
If you
pretty
see one
consistent.
taller, I'll give it a whirl.
If it's
Uh,
Looks like a little tiny eyeball of a sponge. Come in a little more if you want. Oh, okay. Very small. Wonderful. All right. Thanks.
Yeah, pilot, we have a couple of requests here on the line if we can get an estimate on the current.
Okay. Well, I'll do a drift test here.
Thank you.
Let's stow before you get closer.
Yeah, sorry. Just trying to get oriented.
Sure.
If you want to push the bio box and I'll do the craft.
Are we just, maybe we just don't have a perception. We're closer to it than we think we are. Yeah.
Yeah, so it's bigger than the.
It's
bigger than the ROV.
It's bigger than the ROV. If that was a sponge, that's got to be the world champion sponge stock on the planet right there. And
it's huge.
Maybe that's why it's laying down.
Hydraulics off on craft.
Whoa.
Oh my God, that is enormous. I don't know what else it would be except for a gigantic sponge stock.
Settling down.
Wow.
Yeah, look at the base of it.
I don't even see the lasers, they're so small. There they are, right there. Holy cow. Centimeters. Nav, go ahead. Yeah, sure. Let's go ahead and call it something like large sponge stalkers.
So I'm going to get the lasers on it for you here, if one of these angles will work out for me. You can come in partial video.
You want to count that as a rock, Chris?
If it's dead, does it become geology?
But I don't think it will fit on any box.
Yeah, I'm not sure it's going to fit in the box.
Well, could you put it in the arm and bring it back?
Well, then it's going to limit the ability to possibly get a second rock, and that would be pretty awkward. We're looking at, I don't know if you can see the Cirrus view from where you are, but you can get an idea of how enormous this thing is.
Yeah, I'd say over 2 meters.
Can you imagine when you give a dog a femur and they try to go through a doorway?
Yes, that's right. That's a good analogy.
Be sticking out on either end and we'd be running into things with it.
Yeah.
Possibly.
Yeah, it's like a chrysogorgid.
Yeah, it looks like a chrysogorgid.
Can make that a lot better if we're interested in looking at it. That image up better.
Is that good enough, Amy, or would you want it from a different angle?
Closer.
That's good, thanks. Do you know the species by any chance?
Nope. Do you? I don't know what that is. Yeah, we're
I wonder
seeing.
if this could be what we were seeing before. Scott, do you think?
It's.
I don't think it's what we've seen before. The density of polyps
All
is...
right, video, we can come out, I think. Thanks.
I mean the one that
near Oka erecta. That's what it appears to be. A little bit different morphology in that the branches weren't broken near the very base. Sometimes you have these little stubs that look like spinal columns going up on this species. With that one, it looked like it was intact. And that right there could be either a dead stock or one of those giant barnacles. I think it's probably a dead stock.
Where are you looking?
I'm looking down at that white blob on the bottom.
Bottom left?
Yeah, I think
just
it's
the base.
just the base.
Oh, yeah.
Yeah.
Looks like a base. Want to snap in real quick video? Thanks. Certainly a base.
Yeah, base of something.
Let's see maybe a black coral base. It's a dark stem or just dead.
Yeah, not a black coral base, yeah. Let's say that's sponge.
Sponge. Go ahead.
Yeah, with an ophiuroid next to it.
Okay.
Yeah, and you got a little shrimp down there. It looks like it's a nematocarcinus tenurostrus, the long-legged shrimp, and beautiful anemone, which is probably an exosalactus again. We could pass this up, pilot, because we got some good images of this yesterday.
Okay. Okay. Maybe you want to come in on this one here first. Go ahead.
So it looks like it's an arella, I would think, with the possibly down-facing calyces. I don't know. I'm waiting to see what Amy's opinion is here.
Oh yeah.
Yeah, Scott.
Maybe to light up.
And Tina's got something here. It got
Maybe
broken
not.
off there.
Swing the left swing arm right. Port swing arm right. Maybe. Keeping an eye that it doesn't hit the shilling.
So Scott thinks it's an arella also. We got 2 votes for an arella.
Take a little more when you're ready, video. Should light it up a little more soon. There we go. That's nice.
So
Thanks, co-pilot.
it looks like it's got these plates, these spicules that's Narella-like. Let's see, Tina's asking, can you zoom in a little bit closer even, or is that pretty much it?
I'll move up to the ends here. Maybe it'll be better to focus on it.
Okay.
Go ahead, video. Is that full zoom? Okay.
Yeah, Paulip says they're, or Scott says they're angled downward. I would agree. It looks Norella. Well, like.
Yep, going back down. Is there a particular place you'd like?
That looks pretty good. A beautiful video. And now the question is, what Norella?
All right.
Yeah, so Amy threw in there several typically barren stalk and then only a few branches coming up. That still looks like there's too many branches on that one.
Video centered up. You can go in on this one.
And we're looking at where it branches.
Okay, I have a note center screen if we want to put in video. Oh, that's not so much.
Yeah, it's just a...
Oops. That's just a turn. There's the branch.
Yeah, that does not look like it's branching from the node, I don't think. So you have a suggestion to look closer to the base.
Okay. Looking down. Acquiring the base.
And right where a lot of those main branches are coming off, right in there, if we can...
Just trying to get around.
Yeah.
These toe holes are very bumpy, so...
And Scott just mentioned that this is not an Ecanella.
Yeah, I would agree. It doesn't look like it's branching at all from the nodes.
Okay.
So these are one of these deceptive ones where you've got a whirl of branches that are coming out near the base, but it's quite different.
75 degrees.
So
thanks.
He again is defaulting to the Corrado Isis, presumably on the basis that the branches are coming off from the inner nodes, these white regions between the black bands. And he said it may be a B-clad. All
right. Thank you, video.
Yeah. Well, that was good. That was two good animals to get close-ups on.
Lifting off and moving on.
Yeah. Very nice. Thank you very much. Norella Bauer's eye.
Video.
I don't know if Amy or Scott would like to weigh in on that. And the basis for this is the longer branches, the fewer branching points and the longer stems between the branch points. And look at this monster.
The pilot spotted this on a Sirius cam.
Yeah, so this was collected in 2003, but we don't see spectacular colonies like this very often at all. We used to call this the gramophone sponge, and it's a new species, has yet to be described. And the only thing we know about it at the present time is it's in the family Eurydidae, and it's in the subfamily Conolasmatinae, and it's going to be a brand new genus.
Coming off a heading just for a few seconds.
Thanks.
Video, can we go in partial on Sirius, please? Yeah, hold that.
Absolutely. Come on in.
Well, sponges don't get better than this.
Sirius view gives you a good idea of how big this guy is.
Surprising there's not more critters on it compared to the one we saw in 2003. It was covered with shrimp and ophiuroids, as I recall. Do you remember that, Chris?
Yes, I do, and that's a great observation. There's maybe a couple tiny things in that one little osculum there, but this is pretty clean, and it's in beautiful shape. It's very alive, well, and happy. So I don't think Henry up in Vancouver is really taken a very close look at this species yet. I sent him a spicule prep that I did in my lab on this one, and he just took a quick look, but he hasn't done his SCM on it yet to really nail down what's going on. But he is certain that this is going to be something brand new.
Can we come wide on Sirius, please? Thank you. Looks like
now, as far as we know, this is not the animal that used to be attached to that gigantic stalk. The ones that we've seen of this particular species are all fairly short like that. And having said that, there looks like there's one with a longer stalk in the back. But as the one that we just saw, but I don't think so. This one's dead. And perhaps it is. It's just a dead, broken off piece of the one that we just saw. So we could probably skip getting a close-up of this one. And in upper right, you can see a Caulophacus, and it's maybe bending slightly in the current.
Oh, I see an eel, I think.
You saw a fish?
Yep. It's coming in by
Oh,
right.
there you go. All right, Astrid, you got another fish.
Going down.
I'll
sidle around this way.
And I'll bet you know what it is already.
Tether to go that way. Go this way.
You got something very shiny underneath you.
And she nailed it. Yep.
Not sure what that is.
Synaptobranchus brevidorsalis.
Can't tell. Okay, video.
Oh. Still looking for that where the dorsal fin starts. It's a little difficult to see on that guy.
Tilt up, cheat a little. Go ahead, video. I was looking for a dorsal fin here.
And there it is. So Astrid's ID was spot on there. It's Synaptobranchus brevidorsalis. And as you can see, the dorsal fin starts right there, very far on the back. And that's actually the basis of its name, brevidorsalis, or short dorsal fin, I believe. Aha! So when we're done with the fish, you see that. Oh no, that's one of the plumate ones that we collected already. Okay. I think it's going to be that that Chrysogorgid again that we just collected a little bit earlier. So Scott was wondering whether or not it was just branching on one side, and it's pretty clear on this specimen that it is, absolutely.
Thank you.
And Tina's remarking, yeah, it looks like it's the same one.
Okay.
Diva Amon is asking about what time we're going to be leaving the seafloor. What's that?
Thank you.
Oh,
For watch
that's
leads.
a very leggy. Yikes! Ouch.
Yeah. You still happy with 140?
Yeah, another one of these beautiful sponges. And let's go ahead and continue up if we could. We'd like to make a little more distance, and we've got some really great stuff with this one already.
All right. Thanks, video. No, I think we're gonna skip it. Sorry. Unless you'd like it for your highlight.
Might actually cooperate here.
Come on in, video. Oh, yeah, he's turned his head.
Just ask him to increase.
So he has got very, very long filaments coming off of his pelvic fins, and that would probably put him as a coryphaenoides.
Keep the same
And we'll
move,
see
but
Randy
increase
Singers
speed,
has
just
got
as you did.
coryphaenoides. And I would agree. We used to call this Longiceros, and then we, Tomio at California Academy of Sciences backed us off, and he prefers coryphaenoides species rather than going all the way to Longiceros. So since then we've corrected our database.
Did you have other questions?
And normally they do not cooperate like that, so that is gorgeous. And he's got some little parasites on him, it looks like.
You're not going to get much now because he's driving around.
So Brian Kennedy is commenting again that we're not on the conference call. Still.
You're not going to get much now because he's driving around.
Bruce Mundy thinks it could be
for a particular
Longiceros.
reason?
So I think we're all in agreement that it's coryphaenoides.
How much time we have left in the dive? So that would be the spreadsheet on the left screen. You can fill in current time, current depth, time at 50 meters. Yeah, it should be that middle column.
Thanks. All right.
Pilot, you are off to my side.
Thanks.
As you can tell, do you want me to follow?
No, I'll pick up and come your way.
Roger.
Thanks, video. Or is that a biomat? I see a particularly shiny spot. I'm just not sure if it's a glass or a biomat right here. Quick zoom video, if you don't mind. Just caught my eye. It's being particularly shiny. Yeah, go ahead. Can you tell what's going on there?
We get these little patches of the manganese crust that can be quite smooth like that down here.
Yeah, it looks polished.
Yeah.
Wow.
Yeah.
Well, for my own sake, I wanted to take a look.
Sure. Yeah. No, they're not that common. They're really really interesting. Exactly what polishes them like that.
You can stay in video if you want. I've got a good toe hold. Just anything of interest. Just hold it for a couple seconds and we can go. Great, thanks.
We're probably going to survey here for about an hour before making our way up to the surface.
Okay, Dan, thanks.
So far we've seen some pretty nice densities of corals and sponges, although a lot more corals than what we saw yesterday where
Hey, Chris, this is Jonathan. You see that black rock right in the middle of the lighter colored sediment back to the left?
yes. Yes, we do see it, Jonathan.
I wonder if that one is loose in another potential sample.
Okay. So, pilot, we have a request that rock there that's in the center of the screen in that sediment pile. If that is loose and grabbable, they'd like to see if we can collect that sample.
Roger that. We'll get ourselves set up.
Can we please bring the ship to an easy stop here? Thank you.
So, Jonathan, are we looking at the inside of a broken pillow here?
Thank you, Bridge.
Yeah, most definitely so.
So perhaps if that one in front is too big, maybe one of those in the back there might work.
Yeah, I think it would be neat to get a sample where we know it's probably sourced from one of these pillow lava flows.
Okay.
Yeah, the size of the rock is certainly appropriate for collection.
What's that, the big round one?
Yeah. It's about 20 centimeters across.
Yeah. Oh, okay.
Oh, hi, Jeff. Here we are.
Yeah, so
barnacles.
They do look like barnacle scales, don't they? I think they do. Yeah, they. It's our best guess, as these are loose plates from barnacles that have died. Quite a lot of them, but we're not exactly seeing a lot of barnacles in this area. That could account for this graveyard here.
All right, thank you. Yeah. Arm is active.
And so Santiago also thinks they could be barnacle plates as well. He can't tell from the video resolution, but Santiago, that's our best guess as well, looking at a clear screen here.
The larger rock is not yielding.
Third time's the charm. Think you could get that one to the left of the large rock?
Something. What appeared to be a rock there now seems to have disintegrated.
That's not a good sign.
Not sure what that was. Come on down, Jeff.
Am I pulling you around?
You're getting ready to. We're swinging a little bit here.
Okay.
As you carry on, I'll see if I can't make quick work of this. No, that one is unyielding as well.
So where that
What about the
drop
plate
was,
light
is that
rock
just
in
gray
the,
on
on the
top?
right in the back a little bit? Think you could maybe even break a piece, yeah, either that one or the other one? If they're rocks, I'm not. No, that's just coming apart.
Just let the dust settle a moment here. And something back there.
So, Jonathan, what about this flat one he just loosened up?
Ship move,
Will this be
15
satisfactory?
meters, get me back towards.
Copy that. Stand by.
Thanks.
15 meters.
Yep. Dave, I'm going to turn my back to you, just so I stop tugging on you.
That's fine.
Not sure he can hear us. See if maybe ask him over the chat room whether this rock will work.
Dave, I'm going to turn my back to you, just so I stop tugging on you.
That's fine.
Not sure he can hear us. See if maybe ask him over the chat room whether this rock will work. Yeah, we dropped the conference call again. Yeah.
Bridge nav. Can we please move the ship back toward 320 for 15 meters? And we'll go ahead and do that at 0.1 knots. Thank you.
Well, even if I get pulled off, I have the rock. Alright.
Okay. Oh, just broke. You know, because
So,
it's so fragile, I'm not sure they're going to be interested in it. Given that they can only collect 2, we might want to
Thank you.
Go ahead and put that one back and see if we can find.
Alright, we will turn it from whence it came.
Yeah, so Scott also agrees it might just be a chunk of manganese, and so.
Yeah, unfortunately, we've lost him on the conference line, and I don't think Jonathan can hear us.
Yeah, he may not be looking at the chat room. Or is it too close to this
it's
sort of...
definitely an interesting pile of talus. Probably have a better chance of finding something loose around here, if anything. So if you see something that looks obviously loose, then I would say go for it.
Well,
but it could be this broken stuff in this sedimentary here, these layers here, or... I mean, could it just be some of these plates that are broken off?
You mean under this ledge?
Yeah, and all on the bottom.
You clearly see the carbonate layer on top.
I'm not sure what that is. That's awfully flat. Can you maybe zoom into some of those outcrops with the texture?
Yeah, I think he's doing that right now.
Get you set up for a close look.
All right.
Here.
Those almost look like circular top.
Okay, Tara, let's take a closer look, please. Come left a little bit here to pick up the lit section. Oh, Roland. Hi, Rolly. Panning right just a little bit here.
Cliff. That's manganese crust on top.
Yeah, I'm willing to bet that this is more or less a sedimentary layer with that layer of manganese coating the top, making it a nice flat pavement.
So this would not be something you'd be interested in, and maybe a lot of that rubble on the bottom here is probably coming from this type of layer. Is that correct?
Yeah, I think anything we find that's close to it is probably eroding away from this little incised small wall right here. So very interesting feature, just not worth a rock collection.
Okay. So yeah, that's what they look like, barnacle plates.
Yeah, and then Scott's just asking if those are barnacle plates, then where are all the barnacles? And
yeah, that's the big question.
Yeah, I don't know. I have not. I haven't seen them either.
What eats barnacles?
Well, these barnacles are pretty big barnacles. So the really big one that we collected a number of years ago, this is in 2009, and sent to the barnacle guy at the Cal Academy of Sciences. His name is Bob Syak, and he identified, just seeing the dead plates here, but that was the big one down there that was also creating some of those plates, because we saw live barnacles directly above a graveyard like that.
And there's some more discussion on this. Santiago says that he's seen accumulations like... yeah, Astrid, looks like there are a lot of dead sponges here. You can see some of them topped over there with the bases still intact.
Dave, are you
hold
drifting
fast.
up and back because
Yeah,
of partial zoom?
Yeah, I am.
Okay.
Sorry, Roland. Let's come wide now, please.
Just noticing it was very blue.
Yeah.
I wonder if I need to recalibrate.
It's just...
Looks like some interesting commensals there on that.
Let's get a quick zoom
that
here,
coral.
please, Roland.
Some anemones.
Yeah, so both of these are isidids, bamboos, and this one almost looks like he's branching from the nodes. I'm not positive.
Let's get a little more zoom.
Maybe right at a branch point a little further down.
Yeah, maneuvering.
Partial.
That looks like a nodal.
I agree, Chris. I think that's branching from the nodes. It's some kind of isidella.
Yeah.
So have you seen this before, Scott? I don't think I've seen this one before.
I'm not sure. Again, I've got this poor setup. I'll fix it tomorrow where I can't be close to the screen while I talk to you, but I've seen something like that in the Bahamas, and sometimes once in a while you'll find a node where there looks like three branches coming out at an angle, not like an Acanella where they're perpendicular, but where they stay planar, but there's multi-branching coming from a node.
We've reached
Yeah, thank you, Scott.
Copy that.
I can set down for a macro shot if need be.
Sure. Yeah. Why don't we get a whole colony shot? Since
Wide,
you
please,
got a bunch of us stumped on this one.
Dave, do you think you'll be here for a while? Shall we stop
No.
the ship?
No, we don't need to stop the ship. We just need to get some stability in the video. Okay, Roland, I'm bottomed. Zoom at will.
Yeah. Oh, there is also one of those commensal anemones after this that maybe we can get. He was actually facing the right side. Mostly we've been photographing anemone rear ends, so it'd be nice to actually get the front end for a change.
Chris, you've seen more images of Isidella trichotoma. This is not that. Is that correct?
Yeah, it is not.
It's not that from what we think is Isidella trichotoma. The one that we believe is trichotoma is extremely planar, and it's very dense branching, and I'm not seeing that in this colony.
Yeah, no, we saw a couple of those earlier, and this is very different.
But I see what you're talking about. You seem to have several things coming out of one node, in at least some places.
Right, but it's not bushy like Acanella. Those branches aren't arising perpendicularly. So this is what we would call classically Isidella. Genetically, I'm not so sure that's the case, but that's what we would call it for
I'm
now.
going to pull out for you to go wide up there, okay? Just so
Scott,
that
were you watching when we got that real planar one yesterday? It was before we actually got into the higher density area.
I'm not sure which one of those he wanted.
I don't remember which one you're referring to. There was one that looked planar above, but then when you looked at the base of the colony, it was clearly Acanella-like. So I'm not sure if you're referring to that or another one. Sorry.
No, it might have been that
the
one.
bottom one, which is
So,
facing us. Although that top
yeah,
one's
I guess we still have some confusion over who's who down here.
Well, I don't feel like I've seen this one yet, today or yesterday. So this is a really interesting find, and so it's nice to have these images. Thanks.
Yeah, and I totally agree. I'm not so sure I've ever seen this one before either. So this is terrific.
I suppose I have to keep in mind the possibility that could be something like Jasonisis, which has that type of branching. But the scenic just doesn't look quite thick enough to me from this angle.
Looking for that dark red.
So we got both the front and back side of this anemone, which is terrific. If they're the same thing, and there's a good chance they are.
Get this for a few seconds, and I'll just push to the one in the
And
background.
it looks like it's got a textured stalk here, which could indicate it's a type of hormathiid, but I can't really tell whether that's a sort of a chitinous stalk on it or not.
Okay, push into
but
the far
typically the hormatheids have a very rough textured stalk, and that's one of the diagnostic characters. The one above it, though, hard to tell because it's fairly translucent.
And then keep going up. We'll
And
get
I'm
the
sort of leaning toward it not being a hormathiid, so it's probably some other type of anemone.
Beautiful shot of a crinoid there.
Yeah.
Dave, you have to think about moving in 1 minute.
Well, that'll be perfect. Just finish this up here, Rolly. And
yeah, we need to get a full screen after this.
Okay. We'll be all right.
At the polyps very clearly in the
So we are currently at 2,377 meters.
Okay, pulling for full
For
wide.
those listeners
for
that
the
just joined
full
us.
Coral. There.
Got it.
You want the lasers off, Rolly?
That's okay.
Okay.
Not showing up. Okay, I gotta go here.
Full wide.
You happy, Rolly?
Yep. Yep.
Okay, we're out of here.
Thank you, sir.
Straight ahead, Dave.
Look at that.
Bubblegum coral.
So very interestingly, we keep coming up slope, and we still haven't gotten into the bed of the corallium, which is what we found the other day, or the other year in 2007.
But we are quite a bit deeper. Yeah, we're about 600 meters deeper. But it is very interesting to see these densities of corals extending down this depth.
Yeah, I suppose for both the pilot and the nav, that's one of the reasons I'm interested in getting up above waypoint 2 is to see if we can actually yeah, I think it's pretty certain it's going to be a Tritopleura.
Pulling out for the bounce.
Not much I can do about that.
Yep. Once it stabilizes, I'd like to go back in again.
Nice bathy pathis there.
Yeah.
Okay, we're good for zoom here, Roland.
Okay.
It's like a hydroid growing on the sponge.
Just trying to come right here a little bit. In the cobbles.
Pulling out a little bit.
There, just hold there a minute. Let me get.
Can we zoom that crab for Mary Wickstein while you're here?
Okay, rolling.
Yeah, I don't know if you're right there too.
Yeah, just a couple arms peeking around. So I'm fairly comfortable with this being, again, Tritopleura in the family Uncinnatarity. And that's the sponge. And I don't know what the little lobster is, but he could be a Munidopsis. His pincers and his arms are a little bit stubby. But I'm probably wrong on that because it's not quite right. He's definitely not a chirostylid. I don't think his joints are long enough for that. Could be a Munida or Emunida. Got a little, looks like a little gastropod.
Yeah.
Permission to zoom on Sirius.
Go for it, Roland.
The blacks.
Just another clear example here to the general public that might be following why we're interested in these corals and sponges. They do create habitat for all kinds of other organisms.
Very soon.
There's crabs, hydroids,
Pull wide, quick, Roland.
Copy that.
Barnacles, anemones, all kinds of other organisms that grow on these sponges and corals.
There we go.
Pulling out.
This image here, 2,350 meters on a seamount north of French Frigate Shoals.
Looking backwards,
Yeah.
Pulling out wide on Sirius.
Coming right our way.
Snap on him. Swimming
no,
away from you.
I just catch him up. He's inside the light.
There's a crinoid below you.
Very nice mushroom coral there.
And we could get a little tighter on that sponge next to it also.
We're working on it.
That's going to be the smallest baby Tritopleura I think we've ever seen. It's good to see what they look like when they're really young, and that's what I think it is.
The beagle has landed.
Pushing in. Getting a two shot.
Yeah, hang on a second here. Let me.
So we typically go out on a limb here and call these mushroom corals Anthomastus fisheri, and it's not really clear whether they are that species. There's different color morphs. Sometimes the actual polyps have red stalks on them, and we just call those Anthomastus red.
Going to push in
And
very
sometimes
tight
we
to
find these
the
colonies
orange.
with very few polyps, and they're completely white with no red color.
Max zoom.
And we call them Anthomastus white.
Look at the dots. Pulling to the polyps.
So Scott spotted a crinoid. No, he says it's
yeah,
coral. Okay.
Now there was a crinoid on the sponge
looking
that
at the
is
sponge.
closer to the vehicle.
Oh, okay.
Tilt down just a smidge.
Less than a minute, Dave.
Max zoom.
So these are beautiful.
Okay, let's wrap it up a moment.
Okay.
Beautiful sponges with
Pulling
a very fine texture, and if you put some of the sponge in a little bit of bleach so you can actually see their skeletons more clearly, they're absolutely stunning. Sponges are beautiful as they
No
are,
time for that,
but
the
when you see
crinoid,
them under
right?
the microscope and see the amazing structures
go.
that
Copy.
come together to create the whole colony, it's just really thrilling. They have all types of different spicules, and that's what they call these structures that make up the sponge, microscopic structures. And they can come in the form of long needles and spiny type of three-dimensional, axial structures. They can look like balls with spines on them, all types of things. One of the common
long-legged.
shrimp. Yeah, the long-legged nematocarcinus.
Yeah.
Yep, very common at these
Wide,
depths.
please.
Great, thank you.
So,
For those of you who have been with us for a while,
Okay, Rolly, you can zoom.
we landed just under 2,500 meters. Yeah. And
so
Full screen.
yeah, that is
all
a.
right, it looks like it's a corallium.
Yeah.
And I don't know if Amy is on the line here. Definitely
You center it
corallium,
left
so
to right.
good.
I can max it out.
Yeah, working on it.
And so as far as the species goes here, so I think, Amy, if I'm not mistaken, this would fit in your new
giant
genera,
candelabra
I think it was
sponge in the Sirius
hemicorallium, if I'm not mistaken. Or is this unclear?
Ready for max?
Yeah.
Well, actually, you got it really positioned, so we'll let that be
Looks
our
like some
full
of these stalks are
species.
elongate, and I don't see too many clumps
Oh, yeah.
right on the end.
That changed the lights. Okay, going for max. That's max.
It would be really hard to tell without a specimen, I think,
Yeah,
or a sequence.
but don't you want to go out on a limb? I mean, we're throwing out guesses right and left. Because
I would be more in the genus probably corallium with this one because it looks a lot like Valenza, which is stayed in corallium.
I'm
Okay.
passing you by, Dave.
Oh, thank you, Amy.
Okay.
Yeah, thank you.
Ready for full wide. Can we spin right to see that giant
So
candelabra?
we were hoping to actually see some of these on this transect. So maybe
swoop
we're just
by.
starting.
Very beautiful
So-
sponge.
Yeah, this is the backside, I think, of that really beautiful one that we were looking at before. It's kind of nice to see both sides of them. I'm certain it is.
Understood.
So they're just as beautiful from the backside as they are from the front side. And again, this is a very, very healthy colony. Really gorgeous animal. And we'll confirm it's the same thing, and sure it is. There's one commensal on that,
Watch your
that
toe.
one, but again,
Doing great.
there are not very many of them.
Beautifully done. Great
Yeah,
move,
for
Dave.
that size.
That thing is so reflective.
So that was a beautiful little turn right around that colony. That's a beautiful video sequence where you get to see both sides of it.
Heading up the hill.
And so that sponge, for those of you who didn't see the previous one, that's going to be a new genus. It's in the family Eurydidae, and it's in the subfamily Conolasmatinae. And so it does not have a name, and it doesn't even have a genus name yet, because it's a
That is another beautiful
still needs description.
specimen there on the right.
Right now, we
Flyby.
have identified approximately 40 of these deep water sponges in Hawaiian waters that need names. They're all new species to science, and it's just a matter of finding the time and having enough people to actually start describing these. Henry Ricewig up in Vancouver is considered to be one of the world's authorities, if not the world's authority, on these type of sponges, these siliceous glass sponges. And everybody is vying for his time and not allowing the poor guy to enjoy his retirement. So, and Henry works, I worked with him for 2 weeks, and he works about a 12-hour day every single day. And now we've got another one of these possible coralliums, and next to a gorgeous specimen of Poliopogon species B. Being worked on again by Henry right now. He's actually actively trying to make headway on describing some of these sponges, and he's looking at 5 different species of these right at the moment. So my best guess on this particular Caulophacus is Caulophacus subgenus
Got time to
Caulodiscus.
zoom into the interior day?
Yes.
Yep. So pretty much all of the members of this genus also have subgenera names. So there's a Caulophacus caulodiscus, and there's a Caulophacus subgenus.
Okay.
And there's a
start to come in.
Caulophacus
Permission to zoom in on
oxydiscus.
Sirius?
Yep, go ahead, roll.
And then there's a couple that nobody knows what subgenera they belong in, or what subgenus they belong in.
Maybe the laser's off.
Lasers off, yeah.
Right, tilting.
Going for max zoom.
Roger that.
And that's a remarkable look at the spicule
Tilt
structure
down, I think
in
I
the
saw
sponge.
some life.
And you can certainly see it's a
towards the
very
bottom.
complex
Shrimp.
structure, and you can see the claws of somebody hiding in there. Some type of a small, either a shrimp or a small
Or
lobster.
crab, yeah.
Pulling out as we pull away.
Okay.
Gonna hold Sirius until you turn.
Right.
Full wide on ROV.
Lifting off.
Another one of these sponges, and all the ones we've seen of these have been pretty clean of commensals, perhaps indicating that they might have some kind of chemical or structural defense.
Start to see the barnacle.
And there's a stock crinoid.
Stock crinoid. Do we have time? Scan it top to bottom.
And perhaps we could take a moment and take a look at this one if we
can.
could. So this may be the same one that we saw yesterday, or maybe not, because it seems to have a lot of either branches coming off of the stalk or something's growing on it. So you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 arms.
Okay,
I think.
rolling.
Okay, going in for full on the stem.
So something is growing on it. And I don't know what those are. So Scott or Amy are some more of our Nidarian experts. Any clue? Are those...
Those look like little hydroids.
Yeah, I agree with you,
that are
Scott.
growing on it.
Yeah.
So I'm sorry, I don't know if you heard me. This is Scott. Those are little hydroid colonies.
Oh, okay.
Yeah, I agree with you. Yeah, you don't see any branching on those.
I'm not sure I've ever seen hydroids growing on a stalked crinoid before. And after this, if we could get up toward the calyx a little bit, and yeah, maybe take a look down at the base. It's hidden a little bit. And then get up right at the base of the calyx, and we might have a shot at identifying this thing.
And even here, it looks like there's some...
That's a partial zoom
Yeah,
out.
right there. That's a perfect angle right in behind those branches where they spread out.
Blow it, Dave.
It'll come back.
So Nicole just chimed in here and said there might be a Hyocrinidae.
So Nicole, the only hyocrinid we had out in Hawaii that I'm aware of had 5 branches. So do we have hyocrinids that have more branches than that? Because I'm not that familiar with these stalk crinoids.
Let's go back in, rolling.
Yeah, right now we in our gallery we have one species of Hyocrinidae that's been identified to genus as Tylocrinus, and that has 5 arms. And then we have an unidentified Hyocrinidae, and that has 5 arms.
Right.
And this one has quite a few more. So it'd be really interesting if we could expand the number we have in this family to something that's got 8 arms. And that is spectacular. It's beautiful imagery. And this evening I'm going to go into Charles Messing's artificial key
Let's
to
come
the
wide, Rolly. Our time is
crinoids and
growing
see if I
short.
could figure this one out.
Full wide. There's a shrimp in the top. The crinoid just
Straight
took
down.
off.
Guts to go.
So Chris Ma, according to Nicole,
Yeah,
Chris
crinoid
Ma was
fly.
identifying this as
See
a
him.
Hyocrinus. Chris, I don't suppose you're on call right now.
Oh, do we have a Pygnogonid there coming down?
I didn't see that. Now this is what we describe as high
Not
density.
Exactly true.
Yep.
And you're looking
And
at the
this is what I was
0.5 meter
hoping we
contour.
were going to get into.
Yeah.
Yeah, I misread the
One thing you'll
instructions
note
on
is the
making
majority of these
contours.
animals look
Okay.
to be the same
So
species.
The feature keeps going down to the southeast.
And something we
Gives you
are
that little bit
seeing
to see
in
the
these
change
really
in
high
color.
That was
density
the end of
beds
the
is you're
range.
going to
That's
have
the end of the
a
range
few
of
very, very
contours.
dominant
Do you have
species
the
and then
ability
a
to look at those
smattering of
guys on
others
your
that
left?
are
Definitely keeps going up to the southeast.
not so high density in their...
Density's breaking
and,
down on the right.
Scott just... Yeah, that would be worth getting a zoom in.
Yeah, definitely worth getting a zoom on this thing. Good spot, Scott.
Thanks. So that's Stauropathes.
Stauropathes.
Black coral, the genus Stauropathes. Tauropathes.
Yeah, and we're also seeing those toothbrush sponges as well. Let's get a zoom on this thing. Yeah, it's
Okay,
yeah, the Bathypathes was a premature guess on it. I agree.
So yeah, the only species that's described here from Hawaii is the Steropauthis sterocarata, and I believe that's quite a bit shallower.
That is a gorgeous animal. That is beautiful. Any idea what species this could be, Scott?
Once we get this, then I'll go in full.
Hi, Chris. No, Storopathy storocrata is the only one I know from here. I'm pretty sure we collected this specimen that Dennis may have used to describe the species on
So it comes
one of
back
the
up.
hurl dives with the Pisces. So I'm
See
not
the
sure what other
yellowish
ones are
of the
here.
two polyps?
Yeah, no, that
That'll be
Scott,
my
that's the
focus.
only species that isn't recorded from Hawaii.
I wonder if that's a tiny
But
little,
that is
Copy that,
I
Daniel.
think it's just another polyp.
I think he wanted you to repeat it.
beautiful imagery.
Yeah. So, Scott and Daniel, what makes this Steropathes? What are you looking at to identify it as this for everybody's interest?
Pulling to the foreground.
What I'm looking at is the main pinnules that come off that center axis branching. So they're branches off those main pinnules. And the way it's kind of curled over and to one side, we used to call them catcher's mitts. So this looks quite a lot different from the ones that we see in the Atlantic. And I honestly, at this point, don't remember what the Storocrauda looks like. But I'm just about to go into my database and see if I can find
Going to
anything
pull
helpful.
for full
Oh, great. Thank you.
again. Yeah, we can see the very top, the polyps that are turned towards us.
That
Maybe
is something.
A little bit left.
That is just beautiful.
Too bad our collection boxes are maxed out.
That's max.
We can't just collect black corals, Daniel. we're gonna need to space them out a little bit. I think some other coral biologists here might want to have a
Oh, absolutely.
few
Yeah.
other things. I sacrificed that toothbrush sponge for these, but it was a good sacrifice.
There's a
As
sheet
I
there,
think we
that
got some
whiteness
really
in the
great
back.
specimens
Oh, I
there.
see it.
Yeah.
Waiting for it to come into view.
No, I can't give you any more information than just Storopathes, so that's the best guess for now. And one day we'll find another one, and we'll get an idea on it. But thanks, nice close-up.
Yeah, thank you.
Okay. Pulling out.
Dave, I've turned left to look at you.
Roger that.
You're full wide.
There's this.
Oh yeah,
Dark,
some more of the
Do we have
purple
a tank or
crinoids.
purple?
If we could
pop
skip
in on
those
it
for the
real
moment,
quick.
and
Chris, it looks like some loose rock in there.
yeah, they're trying to bump them right now, Mike, to see if any of them wiggle.
Barnacles
Oh,
at the
they're
top of
using
that
the
coral.
ROV to bump it.
Yeah.
I really don't want to bash that one.
Brute force, isn't it?
I'm not really seeing anything loose.
Let's see what else.
In the Nipkins, you can just see how it's coated and
It's yeah.
rounded.
They're definitely trying to throw their weight at getting you a rock. There you go. They're having a go at this one, I think. Almost.
They are uniformly cemented. We will. This is a challenge for our engineering team to develop some parrot jaws or some other mechanical advantage to crack these off.
If there was a rock here that was loose and not cemented, you'd wonder if
There
it's
it is.
Did it move?
I just
Yeah,
found one.
something just fell down.
Oh, he's got a loose one. Okay. That's pretty big.
It comes with a bonus.
But there's a little small one there.
Yep.
It's a very light colored interior, Mike and Jonathan.
Yeah, we're at a
Is that
stop.
something you still might want?
Permission to push in on Sirius, super tight.
I think that thing is just a coating of alteration on the rock. So, and you get a bonus there, I think.
So one of these would be acceptable, one of the ones that moved. Is that correct?
Yes, sirree, please.
What's that? They want the one with the...
Yeah, the one that has...
okay. Will that work? Is that small enough?
We'll see.
Okay. That one could probably go in the open rock box, or it's not going to get confused. If
trying to find the lasers here. And
Hydraulics are up and running.
Can't see the laser spots. Do we have lasers on?
Yeah, I don't think. There you go.
Oh, there they are. Okay.
About 40
It's a big
centimeters. Yeah.
But I'll try to get it in the box.
So, Mike, they're going to try to do it. It's a big boy, but they're going to try to do it.
Why don't you bring these?
Jason has picked up bigger samples.
Uh-oh, the competition has been set
Dave, you want me
here.
to zoom in on ROV in the meantime?
If I'm not mistaken, you just slapped the pilot in the face with a glove, didn't you, Mike?
Starboard wing inboard.
And after
Can you repeat that, Chris?
After he brought such a great specimen.
If I'm not mistaken, you just slapped the pilot in the face with a glove, challenging him.
Was not intentionally.
Hydraulics active.
Well, now you've forced him. He's got to get it in the box now.
Dave, before we move it, should I zoom in on the ROV main HD?
Yeah, you should zoom in. Let's get a good look here.
Can I take your mini Zeus?
You can.
Here, tilt the camera down now.
Okay, tilting. Let's come a little wider.
Definitely got some manganese crust on it.
Feel the screen.
Yeah, the crust looks pretty thick. I hope it doesn't just keep falling apart and they were able to pick it up.
Well, we will find out shortly, I think.
Okay, let's come wide. Great, thanks, Rolly.
Just checking to see if it's alive, Mike.
It's heavy.
All right.
Okay, Roland, I think we can
Oh, it looks like two cemented together.
We've been missing the chat log a little bit. We got so focused on the rocks, but there's quite a discussion about some other animals that folks are seeing down there.
And Amy asked how much more bottom
When you
time,
hold
and
it
it's
there, can we
now
look at the coral?
4:30 local time.
See
We
if
have
it
to
makes
be
it
back
to
on
the
the
surface.
surface by 6, so we probably
center
will
it
leave
up.
the bottom
Okay.
here fairly shortly after we
not
collect
lose
our
your rock,
sample.
Okay, go ahead. It's tilting up. Okay, let's come wide and you put this away before much longer.
So I'm having a little difficulty what we're looking at here, Scott or Amy.
Okay, go ahead.
That's not a primnoid, is it?
You're clear.
It's, is that an icidid? I wasn't really clear what type of polyps those were.
Stoloniferous octocoral.
Okay.
Thanks, Scott.
Yeah, thank you.
Yeah, it looked to me like a stoloniferous octocoral overgrowing,
Co-pilot, can
perhaps
you adjust HD2 to get a better image?
Oh, okay.
some other.
Okay, great. Yeah, I
HD2
wasn't
on the
even
right screen,
thinking that
please.
way. So,
Coming.
yeah, thank you very much.
Great, thanks.
So Randy
little
Singer
bit,
was
please.
wondering whether we're going to do our typical speed run as time runs out before we surface to cover a lot of ground, and I guess that depends on how close we are to when they've really got to lift off, and I imagine we're pretty close.
We
So
might
this is
need
going
bigger
in
specimen
the
boxes.
starboard open rock box, because the thing's way too big to put in any of the closed boxes. Oops,
It's
just
also
broke in half.
friable.
It's still considered one sample, right?
Let the dust clear a little bit here.
Yeah, starboard forward rock box, that's it. Wonder if you could just put both pieces down into the,
Gonna try.
the box. And
I wonder about our weight and balance here.
yes, it is.
Gentlemen, it's maybe time to make a choice here. Let me get some light on the subject. Yeah, which piece would you like, with coral or without coral?
You want to zoom in on our HD2?
So we've got a question here. Which piece would folks like? The one piece is already in the box, and we've got a piece here in the hand. And there's, I think, some concern about whether that would be counting as 2 rock samples, which would push us over our 3 limit, or not. So I think they're thinking of which piece we'd like to go ahead and grab. Is that correct, Pilot?
Yeah, I really only have room for one here.
Okay, then why don't we put this one down that's in your claw, and we'll just go with the one that's in the basket, and then there won't be any conflict with how many corals we took either.
Excellent. That's
Chris, I think the one that you just dropped in the bucket, the one or the piece that broke off that's in the bucket, is actually the one that we would like to have. So
Yeah, that's what we're going to do, Jonathan. We're going to keep the one that went into the bucket.
Okay, thank you.
Take a nice view of that one, though, since you faced it so perfectly. You see the interior.
There you go, Roll. You can zoom in on that.
On HD2? Or
Is
it's
that
one?
what you wanted?
HD1. We can do both.
Oh,
but
sorry. I misunderstood. Never mind.
Great job, guys. Thank you.
Yeah, you're welcome, Mike. I'm certainly delighted that we were able to get you folks your two allotment of rocks there. And it looked like it was getting pretty close there, whether we were going to get that second one.
Okay, I'm going to swing my arm inboard a little bit for stowage.
Okay, and I got the bio boxes all set up to open.
So watch lead, we still have 15 minutes until off bottom.
Dave,
All right, let's make a run for it. We have 15
rock.
minutes. Let's go up slope. And,
Finished with HPU.
All thrusters full ahead.
Hydraulic secure.
Getting a little close-up of the broken edge. Just look at the texture there before we leave.
Okay, we're clear, Dave.
Okay. Let's come wide.
So pilots, I'm going to, if it's all right with you, I'm going to put in a request for 80 meters, continuing at 135, 0.2 knots. That'll take us to the end of the dive.
We're okay with that.
Sounds good. Dave, I'm looking straight down.
Bridge nav.
Michael, can you explain to people watching what all those colors might be and that rocket shoot
I'd
off?
like to request an 80 meter move at 1.3.
Yeah. And a shadow.
Oh, yeah.
I swung it inboard a little bit more than usual. So my intention here, Jeff, is to let you catch up to me. And,
Yeah, I was assuming
get
that,
your
so
light
thank you very
pool
much. That
on
would
the
be
wall
terrific.
there.
So what they're doing is they're waiting for Sirius to...
Get on that fish.
yeah.
So they can rotate this way.
Yep, don't want that rotation.
No. So I can rotate them the other way. And that's as
So,
far as they rotate.
Randy, with those long pelvic filaments, would that put it as another Coryphaenoides, perhaps a small one? The head doesn't quite look right for Coryphaenoides.
Yep, that's what he thinks it is.
Coryphaenoides, okay. So this is a younger one, and he's got quite the little tag along on his side, doesn't he? Yeah, so maybe you can move a little further upslope and see if we can get one last shot of the community.
Like a commensal on the back.
I'm going to need the mains back on to do this.
You see a crinoid on it.
Stand by a moment.
Looks like a starfish.
So I don't know what you're thinking here, Amy. This could be a Paracalyptrophora in my mind. I know they're on this site further up. But I'll be interested to see what you and Scott have to say about this one.
Let me reposition. That's not.
Yeah, if you're going to position off to the right.
Yeah.
Looks like your tether's pretty straight though.
Yeah, so Scott was sort of mentioning that you don't usually see these sea stars feeding on the corals, but it's not really clear whether it's feeding on it. It may be, or it may just be an ophiuroid. It kind of looks like an ophiuroid just sitting on it.
Yeah, that's what I think. From this angle.
Take a little zoom, Roland.
Yeah, definitely an ophiuroid. Ophiucoma, I believe.
Oh, we've got an ID? Is that right? Or is, okay.
Turn it left to look at you.
Now that's beautiful of that Ophi.
Getting a little tether bounce here.
Yeah, pull out for medium shot there.
So we also have a crinoid over there, one of the white comatulids, looks like.
Pilots, watch lead. Once we're done looking at this coral, we're going to have to pull off bottom.
You want to look tight at the open.
Roger that, pilot.
Crinoid. Then we'll get a full screenshot.
Nice crinoid.
So does anyone want to take a stab at this primnoid? I mean, I threw out Paraclepifora, but anyone else have another opinion? And that's mostly on the branching pattern and the size of the colony. That is a pretty darn nice forest shot right there. Yes, that works very nicely, pilot. Oh, sorry. So, so video, yeah, that, let's see if I can work this thing. Yeah. Yeah, so, yeah, that is an absolutely gorgeous forest shot. That's the money shot I was hoping for.
So, Dave, you just want to sort of lift off and come up.
That would be fantastic.
And I'll just keep brightening it up as you get higher.
Well, you see those two sponges, and that's a perfect example of the different types of...
Your mic's not open, and you're not talking OKEX.
Did you... Scott mentioned that this is a great dive, and I absolutely concur.
All right.
I couldn't have gone better in my mind. We got beautiful close-ups. We got all of our sample quotas. We made our waypoint objectives, so we got into the upper community, the high-density community.
Oh, actually, it does.
So let's hope we can keep it up.
Looking down. Okay. Fishies.